Have we lost him?
Yeah.
Oh, that's a good start.
This topic sits at the extraordinary
We
crossroads
lost
of
you for
genetics,
about 2 minutes there, mate.
molecular biology, and clinical embryology.
We lost you for about 2 minutes there.
As the science of possibility, the discipline that turns biological uncertainty into informed hope. I always like to tell young professionals entering into the field of assisted conception that we are custodians of beginnings. As every dish under a microscope holds a story that has never been told and will never be told again. Gene embryology is the language through which we listen to that story, and is increasingly becoming the tool through which we help to write its next chapter responsibly. To discuss this topic effectively, we've had to get speakers from various parts of the world to share their experiences. The first speaker is Laura Gaddi, and she'll be talking about genetic testing in embryology, insights into genetic screening for monogenic diseases and aneuploidy. And the second speaker will be Toluwanimi Balogun, who will be talking about non-invasive PGTA, a novel perspective. And then we have Professor Darren Griffin, who will be talking about ethical considerations in genetic testing, balancing technology with patient care. And the last but not the least speaker is Lyndon Miles, who will be talking about time-lapse imaging and artificial intelligence, revolutionizing embryo selection and implantation success. Unfortunately, Lyndon is dealing with a family emergency now. He has promised to try to join. If he's able to, fantastic. But if he's not, we just wish him and his family the best of luck during this period. So I think without much ado, I'll just start the ball rolling and introduce the first speaker so that we can get about the business. Like I said, Laura will be speaking on the topic genetic testing in embryology, insights into genetic screening for monogenic diseases and aneuploidy. She was trained in molecular biology. She obtained a PhD in molecular medicine at Padua University in 2020. She's been working in the field of reproductive genetics since 2015, first as lab manager of the Genomics Italian affiliate, responsible for the PGD diagnostic services, and later as supervisor of research and development department. Our main research is focused on the development and validation of innovative technologies for pre-implantation genetic testing, together with basic research and clinical studies regarding aneuploidies and mosaicism in trophoectoderm biopsies at blastocyst stage. Currently, Laura is the head of scientific support department at Genomics, where she's focused with a team of scientific advisors and genetic counselors in supporting clinics and patients adopting reproductive genetic services. So I yield the floor to Laura to talk to us about this very important topic. So you're very welcome, Laura.
Thank you. Thank you very much for the introduction, for the invitation. It's a real pleasure.
You're muted.
Can you hear me?
Yes, we can hear you.
Okay. So can you see also my screen?
Yes, I can.
Okay. So I was saying thank you very much for the invitation and for assigning to me this topic. I will try to cover as much as possible in these 20 minutes. I will start with PGT definition and aims. Then we will go through briefly to PGTM methodologies and evolving indication. I will focus for the vast majority of my presentation on PGTA, both from the technology perspective and then again the evolution, new things that we can detect nowadays like ploidy abnormalities, and then I will close with just a snapshot of synergies between time-lapse and PGTA. But without further delay, let me start this presentation. So I'm not able to advance. Okay, so according to the definition, PGT can be considered as the earliest possible form of prenatal diagnosis. I hope you are familiar with this term. Usually is considered as a test performed to analyze the DNA coming from oocytes or embryos, or for HLA typing for different conditions, or for in case of PGTM for determining genetic abnormalities. There are many indications for PGT, but in general, they can be, let's say, categorized in two main areas. The one category is inherited conditions, like PGT-SR or PGT-M. This means that we can detect the alteration in parental DNA, and this alteration, either a chromosomal rearrangement or a single mutation or combination of mutation, predispose the couple to a defined risk of transmitting the condition to the embryo and to the newborn in the future. The second category is the one defined or categorized as de novo alterations or de novo conditions, and the difference is that they originate in the gametes, so they are not specifically inherited from parents. We will see, I'm referring mainly to aneuploidies and ploidy abnormalities. But before jumping into the details, let's talk about the process. We know nowadays is quite straightforward. It's a multi-step procedure. You can see we have in vitro fertilization, we can take the biopsy, we collect the biopsy, we send to the genetic lab, and then we have the results. And according to the results, we can take our decision of vitrification and transfer. I believe to maximize the benefit for our patients, we should have close communication between the IVF lab and the PGT lab. Why? Because from one side, we collect the genetic, we collect the material, and on the other side, we're going to process these samples. So this pathway should be optimized to try to get the best possible samples, both in terms of quality and quantity of the DNA. Now, let's go ahead and talk about, I would say briefly about PGTM. It stands for pre-implantation genetic testing for monogenic conditions. The main objective of PGTM is to prevent the transfer of those embryos affected by a specific genetic condition. This means that the couple that we have in front of us is a high-risk couple, typically for a single gene condition. So this means that they have a personal or family positive history for that specific condition, or maybe they can be carrier of a combination of mutation for a recessive condition. So in any case, they have a risk in transmitting these conditions. So we want to specifically identify these at-risk embryos and deselect them before transfer. Now, one of the challenges of PGTM has always been the low quantity of the DNA or the low quantity of material from which we are starting. So obviously, starting from a trifecta biopsy, all the methodologies are based on an initial step of amplification. Could be target to a specific location in the genome, or could be a whole genome amplification. And then we have further step like PCR, could be multiplex of genetic markers plus the mutation. We can have SNP array, we can have next generation sequencing. But regardless of the technology, the principle behind PGTM is always haplotyping, so identifying the mutation or mutations and a series of markers which are close to the mutation, which are and for this reason they are inherited together in the embryos. Now, for many years, historically, this technology has been performed through amplification of the pathogenic variant plus these flanking markers. So the co-amplification of genetic markers and the specific mutation. We can have both direct and indirect mutation analysis at the same time. We have different type of markers, can be STR markers, can be SNP markers. They are different in terms of, I would say, informativity, also presence in the genome. But again, the principle behind is always looking at the haplotype that contains the mutation and those markers that segregate together with the mutation. I will not focus so much on this technology. They are still used and still valid, but in the evolution of the technology, more, I would say, universal methods for these linkage analysis are preferred now. We can have, for example, SNP array platforms where the number of markers that we are in the array is very, very high. So this is a really high dense SNP platform. The principle again is always the same, is a linkage analysis, but is genome wide, so is more extended. There are many advantages of moving from targeted approaches to more genome wide approaches, including, for example, testing for different conditions or skipping the phase that we usually call as pre-PGTM. So we can really use a single platform with many markers inside and test for different conditions. And obviously the protocols are that we typically customize for each family are not needed anymore. The principle behind karyomapping, which is one of the main, I would say, evolutions in terms of PGTM, is to look at the parents and analyze the parents together with a reference. So this means that we know exactly the status, the genetic status of this reference, and then we can assign the high risk or the low risk haplotype in our embryos, comparing with the status of the reference. This is just to show basically the current pathway is very automatic. I would say is also coupled typically with specific software for the analysis. So everything is very easy to follow and to get at the end the final result. Now,
Lara, should we be seeing your slides, by the way? We don't see any slides.
You're not seeing the slides?
No.
Oh,
Sorry,
I'm seeing it. I'm sorry, Professor Darin, I'm seeing the slide.
I'm not seeing slides at all.
No, the slide is quite visible, I think.
Not to
Maybe
me.
check. Yeah, please check. There must be something local.
Okay, not to me. Sorry, Lara, I didn't mean to interrupt you, but I've got
no problem.
Yeah.
So you were able to see the slides or no? Yes?
No, I can't see slides at all. Oh, I can now, yeah.
Okay.
Yeah.
Okay, good. So the other aspect that for me is interesting to explore in terms of PGTM is the evolution of the indications that we have for monogenic conditions, because historically we started with single gene disorders where the genotype phenotype correlation is very clear, and now we are moving more to complex indications. Here we have like late onset disorders, we have predispositions to cancer. Now we're even talking about not only about monogenic conditions, but also polygenic conditions where we have multiple genes that are involved in the disease. So clearly we are evolving in terms of indications, but I believe this is a real challenge in terms of ethical consideration, and we are for sure adding complexity in the interpretation of the results. So we have this special indication for PGTM, which is the perfect technology to resolve all the complex cases. You see cancer's predisposition, late onset disorders. We can have even mitochondrial disorders, which are quite complex to be analyzed, HLA typing. I don't believe there is one single methodology. Probably we still need the combination of more technologies to be able to accommodate all these different conditions. However, it's also true that recently we are moving to whole genome sequencing also for monogenic conditions. So these potentially could open the possibility of analyzing not only many conditions at the same time, but also many indications. So we can have, again, PGT for different conditions. We can look specifically at direct mutation analysis. We can perform PGT for structural rearrangement and also overcome some of the historical limitations, like distinguishing between the balanced embryos and normal embryos. We can run aneuploidies at the same time. Obviously, we have to pay attention, because when we validate new methodology based especially on whole genome sequencing, the possibility of having false positive and artifacts is also very high. So it's important to properly validate these methodologies and pay attention on not prematurely adopting methodologies that, again, can reduce the possibility of using embryos due to false positive results. I would like to switch now to PGTA, because for me it's more interesting as a topic, and also in terms of evolution is more. Sorry, I'm not able to advance with the slides. In terms of the evolution is more interesting, but also challenging. And here you have an overview of the aneuploidy spectrum that we have in preimplantation embryos, which is usually composed by a variety of chromosomal abnormalities. It's a very simplified scheme, but as you can see, we have some alterations which are very well characterized. We have aneuploidies like monosomies and trisomies. Then we have ploidy abnormalities involving the entire chromosome set, triploidy and haploidy. I'm saying they're very well characterized because we know that they are associated with female age. We know the mechanism of origin. Typically, they are uniformly distributed among the embryos. So a single biopsy is typically not posing challenges in terms of predictivity toward the rest of the embryo. But we also have like other intermediate categories or sort of gray zone in preimplantation genetic testing. We have whole chromosome mosaicism. Usually the most common form is mixture of euploid and aneuploid cells. Here we don't have female-age correlation. We still have uncertain and unknown clinical outcomes. So I would say is one of the gray zone where we have to pay attention in not overestimating, not overcalling, but it's still a real biological event in our preimplantation embryos. We also have segmental or partial aneuploidies, which for me, they are the real mosaic embryos that we have in our PGT programs. And in medical genetics, they are called as copy number variants, and they are associated with a wide spectrum of phenotype. We have embryonic lethality, miscarriage, or even genetic syndrome. So this is just to refresh what we can find in our aneuploidy spectrum. Those alterations that are very well characterized are whole chromosome aneuploidies, and usually they are of meiotic origin. So we know that vast majority are coming from an error in the female meiosis, so they are coming through the oocyte. This is important because, and to stress this concept, because these are the type of alteration that we specifically want to target and deselect for with preimplantation genetic testing, because they are associated with known unfavorable outcomes. So when we transfer these uniform meiotically derived aneuploid embryos, we have implantation failure, miscarriages, or even the birth of a baby with an aneuploidy. How we detect these aneuploidies? Typically, we count, very simplified way. We count the number of chromosomes and we say the embryo is euploid, so absence of extra or missing chromosomes, or the embryo is euploid, so trisomies and monosomy. And I'm saying count because the vast majority of platforms nowadays are based on copy number analysis and typically next generation sequencing platforms. Here we have some advantages, but also some limitations and disadvantages. One of the advantages for sure is automatization. When we receive and we analyze PGT samples, typically they are in batch of samples. There are many samples at the same time, up to 100 of embryos are analyzed together. So automation is very important. Also, algorithms for calling aneuploidies are important to avoid the subjectivity of the operator when evaluating the profiles. But what I really believe is important, again, for our patient and to get the most benefit, is to work with very well validated platforms to be able to distinguish between technical artifacts and also real biological defects. Why I'm telling this? I'm telling this because with the adoption of next generation sequencing, we have improved resolution toward other alterations. So we can now see not only euploid or aneuploid embryos, but also small pieces of chromosomes deleted or duplicated called segmental aneuploidies. And we can also see these intermediate chromosome copy numbers, which now are associated with the presence or the presence of euploid aneuploid mosaicism. I'm saying that this is also posing challenges because, for example, mosaicism is one of the most controversial topics we have in our field. So we also need to understand clearly what we are reporting and which is the clinical utility of these findings. Now, going ahead with other information regarding the aneuploidies that we typically detect, this is a distribution of the results that we very typically detect with NGS, but if you change the technology, the distribution is not so different. So this means that regardless of the technology, again, we have the same findings. For example, chromosome 15, 16, 21 to 22 are those chromosomes which are typically always affected in our PGT programs as they are related to advanced female age. We also have other abnormalities like segmental aneuploidies, the one that you see in green in this graph. They are not associated with maternal age, so typically they don't increase as increasing female age, but there are sort of sex correlation also for segmental aneuploidies. So we found that typically a small piece of chromosome deleted or duplicated is mainly coming from errors from the paternal side, while the vast majority of whole chromosome aneuploidies, so a trisomy or a monosomy of the entire chromosome, increase with female age, so they are coming from the maternal side. Now, to really understand the clinical significance of what we are reporting, is important to highlight how much or in which extent a single two-factor biopsy is representative of the rest of the embryo. And a typical study design that investigates not only the cell division origin, but also the predictivity of a single biopsy toward the rest of the embryo is called a multifocal biopsy design or embryo disaggregation model. So working with embryos donated for research, we can disaggregate the embryo and we can follow the propagation of the result starting from an initial two-factor biopsy. And it's nice to see that when we talk about whole chromosome aneuploidies or uniform whole chromosome aneuploidies, this concordance rate among the different pieces, here you have a typical trisomy 21, is higher than 98%. So typically only in less than 2% of cases, we have a different aneuploidy pattern starting from an initial biopsy having a trisomy 21. And this is this means that obviously the predictivity or the predictive value when it comes to whole chromosome aneuploidies for the rest of the embryo is usually very high. So when we detect aneuploidies with very well validated platforms in PGTA, in more than 98%, this alteration will involve the inner cell mass. When we talk about segmental aneuploidies, for example, the scenario is completely different. We have around 30% of alteration which are uniformly distributed, so likely of meiotic origin, but we also found that around 70% of alteration are not distributed in a uniform way. So these are an evidence of a pattern of likely mitotic or mosaic origin. So we have this dual origin for segmental aneuploidies. And when we have multiple biopsies, it's also interesting because we can have more information regarding the type of alteration. So what is really clear now is that segmental losses are more likely to be confirmed in a second biopsy and in the inner cell mass. So this means that when you have segmental alteration in your PGT reports, a segmental deletion is more likely to be true finding versus technical artifact. Now, last step of validation or last step of studies that we have to pay attention when working with aneuploidies is understanding the predictive value in blinded non-selection studies. So typically we transfer the embryo without having the information, and then we look at the outcomes. And this is important because we are not only able to detect Y chromosome aneuploidies, apart for some technical limitations, but when we transfer aneuploid embryos, typically these embryos never give you an healthy baby. So the lethality rate in the majority of studies is 99%. We did the same for segmental aneuploidies. We are starting transferring also these embryos. And again, the scenario is quite different compared to Y chromosome aneuploidies. We have 24% of live birth rate. So this is just to say that segmental aneuploidies can result in healthy babies, but the rates are significantly reduced compared to euploid embryos. So we have to pay attention, because this is really an intermediate category that should be managed in a different way. Same study design applied to mosaic embryos. This is the selection study we published in 2021, but now there is another study confirming the same findings. When you have typically a low mosaic embryo, the transfer outcomes are not significantly different compared to the euploid embryos, and this is both for miscarriage rate and even live birth rate. So I believe that for Y chromosome segmental and mosaicism is quite clear now, and we are now reaching sort of consensus in terms of how to manage these alteration. As I was saying, there are also limitations especially in NGS-based methodology. One of the typical limitations until a couple of years ago was the detection of variation regarding the complete chromosome set, talking about triploidy and haploidy. And considering the importance of identifying these abnormalities, different strategies have been proposed in the literature, incorporating an additional layer of information, which is provided by single nucleotide polymorphism markers. The same that we can use for monogenic disorders are now helpful also for having an idea regarding the complete set of chromosomes we have in our embryos. We did it with next generation sequencing, for example. So is a sort of target analysis inside the regular PGTA workflow. And basically what we want to analyze is the B allele frequency of these SNP markers that we incorporate in the regular PGTA workflow. Why so important? Because haploidy and triploidy, which are these two configurations, haploidy is typically loss of heterozygosity, triploidy is an additional chromosome set, but both these configurations are compatible with embryonic development. So you typically do not have embryo arrest if you have haploidy or triploidy. And even more importantly, triploidy is found in up to 10% of all the chromosomally abnormal spontaneous miscarriages, and usually is not compatible with development to term. So now we really talk about genetics and embryology. So this is really where embryology meets genetics, because historically was the embryologist doing the ploidy test or the ploidy analysis. How? By looking at the number of PNs. So two PNs is equal to normal fertilization. So ideally, balanced contribution from the gametes and the diploid embryo. And then we have a series of typical fertilization patterns or typical PN patterns associated with the presence of one set of chromosome or three set of chromosomes. So it's interesting to talk about the correlation of pronuclear status and the genetic status, because there is no perfect association. So we are now moving to the concept of molecular fertilization check, where the genetic analysis is really able to support and confirm the pronuclear status that we have during fertilization check. So going to the results, when we analyze 2 PN embryos, we know exactly now that 1% of them will have ploidy abnormalities, where the most frequent is triploidy and the less frequent is haploidy. And ploidy is also increasing with, especially triploidy, increasing with increasing female age. So we now progressing in the understanding also of ploidy abnormalities, and we have also, in my opinion, another clear indication for PGTA when it comes to advanced maternal age. To understand, again, the benefit of the application of the test, let me show you some results that we produced a couple of years ago regarding the correlation of the pronuclear status and diploidy status that we provided with PGTA analysis. This is global overview, both static and time-lapse observation. There are many diploids here that we can save. So we should not automatically discard all the atypical patterns that we see, especially if this is done with static observation. With time-lapse observation, the predictivity toward diploidy status is significantly increased. So is it worth probably just to keep in culture and test just 1PN and 2.1PN embryos? And this is also supported by clinical outcomes that we are collecting so far. And this is sort of comfort level in using and transferring these embryos, which is perfectly in line also with the Istanbul consensus that is opening now the possibility to test 1PN and 2.1PN embryos and to go to a sort of cautious clinical use of these atypical patterns that before were totally discarded. I want to close with just this image. It comes from a paper that has very, very recently accepted, so not published yet. And Professor Griffin and me had the pleasure to work in this paper regarding the synergies between PGTA and time-lapse. There is no need to put the two methodologies in competition. They should be integrated at many different levels, especially at the technological level, but also when it comes to the results, because the combination of the two tools is really helpful in improving not only the workflow in the laboratory, in embryology laboratory, but also in improving the clinical outcomes, and also going back to one of the first slides that I presented, in optimizing the biopsy sample and the PGTA pathway. So you can see here we identified many of the synergies that are maybe used every day in the embryology lab, but that for sure will represent the future of the genetic testing with embryology together. So I would like to thank for your attention, and then I will stay for the Q&A session.
Thank you so very much, Laura, and it's been a wonderful paper that you presented. Yes, please stay around for this Q&A. I think we just go straight to the second speaker, Toluwanimi Balogun. Toluwanimi is a clinical embryologist at Nordica Lagos with over 6 years of experience in active embryology practice, specializing in core areas like andrology, ICSI, cryopreservation, pre-implantation genetics, and quality control in ART. She holds a master's degree in the biotechnology of human-assisted reproduction and embryology from the University of Valencia, Spain, and she has an undergraduate degree from the University of Lagos in cell biology and genetics. Currently, her work focuses on active clinical embryology practice and training, while also exploring innovative prospects to improve reproductive outcomes and patient care. So I yield the floor to Toluwanimi to talk about non-invasive PGTA. Thank you.
Okay, thank you very much, sir, for the introduction. And good day, everyone. Thank you for joining us today. And I'll be speaking on non-invasive pre-implantation genetic testing for aneuploidy. So during the course of this session, we will be exploring the principles behind NIPGTA and how it differs from the conventional PGTA and its emerging role in assisted reproductive technology. So whether you're a clinician, an embryologist, a researcher, a student, or an enthusiast, we hope that this session will provide valuable insights into this evolving area of reproductive medicine. I'll also be taking this opportunity to wish all clinical embryologists that are connected online a happy World Embryologist Day. So I'll be working with this outline for my presentation. So what is NIPGTA? NIPGTA stands for non-invasive pre-implantation genetic testing for aneuploidy. So just like the name implies, it's a non-invasive approach for pre-implantation genetic testing for aneuploidy. This enables us to screen or test embryos for the presence of aneuploidy with a non-invasive approach. So one might want to ask, is there a reason why we should screen embryos at all? Well, yes, this is because chromosomal abnormalities or aneuploidy, which is a missing or extra chromosome in an embryo, is the leading genetic cause of implantation failure and miscarriage. Also, as much as possible, we want to improve IVF outcomes, and screening embryos before they are being transferred is one of the ways that we can also increase the chance of a successful pregnancy and lower miscarriage risk. So with the emerging non-invasive methods, we can analyze genetic material from the spent culture media, and we can also aim to remove the process of embryo biopsy altogether. So let's go down to the basics. What is aneuploidy? Aneuploidy refers to the presence of abnormal number of chromosomes in an embryo. And what this means is that the embryo has either a missing chromosome or an extra chromosome, and this can disrupt normal embryo development. It can reduce implantation success. It can increase the risk of early miscarriage and also lower the likelihood of live birth. Just like the image I have on my screen, we could have monosomy, which means that there's a missing chromosome, or a trisomy, which means that there's an extra chromosome. So when we talk about genetic testing, that's the PGT-A for aneuploidy, we could go through the invasive approach, which is a conventional method for pre-implantation genetic testing. And this method requires that we carry out a biopsy on the embryo. This means that we are going to be taking out about 5 to 10 trophectoderm cells from the blastocyst by biopsy, and then we analyze those trophectoderm cells to get information about the embryo. However, there's also a non-invasive approach, and what this means is that we're going to be taking out the need for biopsy, and what we need to carry out this genetic analysis is the spent culture media, that is the media where the embryo has grown in. That particular media can be analyzed. We can get DNA of the blastocyst from that media, and that can give us information about the embryo and let us know whether there's a presence of any aneuploidy or abnormality in the embryo. So firstly, we were talking about the conventional method for PGT-A testing, that is the invasive approach. This requires that we take a biopsy from the embryo. So on my screen, I have the picture of a blastocyst, and this also shows the process of the embryo biopsy being done. And then that cell that is being taken off from the embryo is analyzed, and then we can get information about the genetic makeup of the embryo. This particular method has a very high diagnostic accuracy. However, there are also some limitations. It is quite invasive, and there's that risk of damaging the embryo or damaging the trophectoderm cells. It is also highly skill dependent, and what this means is that it requires highly trained embryologists to be able to carry out this process of embryo biopsy. There's also a link to possible embryo stress during the course of carrying out this procedure, and usually it's not recommended or suitable for poor quality blastocysts. So delving into our topic for today, non-invasive PGT-A. The non-invasive PGT-A is a non-invasive technique that identifies the best quality embryos to prioritize for embryo transfer by screening for the presence of aneuploidy, but without the need for embryo biopsy. What this means is that we're not taking a part of the embryo. However, we're taking the spent culture media, that is where the embryo has developed. We take the media and then analyze using next generation sequencing to analyze the chromosomes and get information about all the chromosomes and find out if there is any aneuploidy or abnormality in the embryo without having to touch the embryo at all. So basically, we're reading what the embryo leaves behind. No biopsy required. It's gentler by design when you want to compare it to the invasive or the conventional PGTA. So this DNA that we'll be analyzing from the spent culture media, where does it come from? How does the DNA get into the media? So studies have shown that as the blastocyst expands and develops, mechanical pressure on the trophectoderm cells drives DNA shedding. What this means is that as the embryo is developing or as the blastocyst continues to expand, it sheds some of its DNA into the media. And so by analyzing that media, we can get DNA from the embryo and analyze this to get more information about the chromosomal makeup of the embryo. Timing matters. So usually we would want to culture the embryos up until day 6 to at least be able to get sufficient representative DNA for this analysis. And also, studies have shown that the cell-free DNA found in the media reflects information from both the trophectoderm and the inner cell mass with a very high concordance. So let's go through the step-by-step process of how PGT works. So basically, the first step is to collect the spent culture media. We amplify the DNA, and then we carry out sequencing to screen all the 24 chromosomes using next generation sequencing. And afterwards, we rank the embryos based on the level of euploidy and prioritize the best embryos for transfer. So let's take a look at the workflow for NIPGTA. So if you want to carry out the NIPGTA, the good thing about this method of genetic analysis is that it's also suitable for conventional IVF cycles and ICSI cycles as well. So if we're doing a conventional IVF cycle, we can go ahead to do our usual insemination where you introduce the sperms to the eggs in the dish. And then on day 1, you carry out your denudation and you ensure that you wash. Now, the wash steps in NIPGTA is very important because we want to eliminate any contamination from either the maternal or the paternal DNA so that we are sure that what we are analyzing is the embryonic DNA. So we carry out our extra washes on day 1 and we can have the standard culture until day 4. However, at the morning of day 4, we carry out individual washes on each of the embryos and then proceed to culture them separately, that is individually in different droplets. And then we leave them in this special culture from day 4 to day 6. And then on day 6, as against carrying out an embryo biopsy on the embryo, all you need to do is to vitrify your embryo, that is to freeze the embryo, and then aspirate the spent culture media for the genetic analysis. This is also similar to when you're doing an ICSI cycle. So for an ICSI cycle, you retrieve the eggs and then you denude the eggs. You go ahead to do your ICSI and you can have your standard culture till day 4. And then at day 4, you carry out the extra washes and then culture the embryos individually till day 6, and then the same process applies. You freeze your embryo and then you aspirate the spent culture media for the genetic analysis. So when we have carried out the process of the NIPGTA, we could have the report come in different forms. So when the report comes back as normal or euploid, that means that no imbalances or aneuploidy was detected in the medium. And as such, that embryo is normal or euploid, meaning that it has the right number of chromosomes that it's expected to have. We could also have a case of trisomy or monosomy. This means that there is either a loss of chromosome or an extra chromosome present in the embryo, and such embryos are not suitable for transfer. We could have a case of no DNA detected. What this means is that we were unable to get enough DNA from the medium to get a definite information about the embryo. And it could also be non-informative. What this means is that a highly reliable result could not be achieved with confidence from the analysis. And then, with this result, it guides us on how to prioritize our embryos for transfer. So yes, we know that the conventional PGTA has been in existence for a very long time now, and definitely has also yielded a lot of results. However, there are still some advantages of this novel method, the NIPGTA, over the conventional PGTA. The NIPGTA is truly non-invasive. What this means is that there is no physical contact with the embryo at all. We're not taking a biopsy, we're not micromanipulating the embryo in any way. Thus, we protect the viability of the embryo. There's a possible risk that the conventional PGTA can pose to embryo development. However, with NIPGTA, this particular risk is completely eliminated because we're not carrying out any process or any procedure on the blastocyst. There's also a broader applicability with NIPGTA because sometimes when you have poor quality embryos that are not suitable for biopsy, we can still carry out NIPGTA because all we need is the spent culture media and we're not micromanipulating the embryos in any way. Better patient acceptance. This is because ethical concerns of biopsies are eliminated in this case, and also less skill intensive. For NIPGTA, it's expected that only highly skilled embryologists are able to carry out this technique. But with the non-invasive pre-implantation genetic testing for aneuploidy, there isn't really a need for that particular reason. Okay, so let's also take a look at NIPGTA versus artificial intelligence. So yes, we know that AI has also helped a lot in embryo selection for transfer. However, with AI, we're only utilizing the image of the embryo. It standardizes the embryo grading using images. But with NIPGTA, we are getting a direct genomic diagnosis. What this means is that the embryo is actually analyzed and we have genetic information about the embryo. And nothing beats having genetic information about the embryo because that can help you prioritize your embryos properly for the embryo transfer. And NIPGTA is also representative of the cell-free DNA from the embryo. So it reflects the trophectoderm cells and the ICM as well, not just an image. Because with AI, you cannot read the chromosomes. You can only use the images that have been generated for you. And so with NIPGTA, we are working with a true molecular result and not a morphologic prediction. So does the NIPGTA have any impact on clinical outcomes? Well, yes, from a study conducted by Sakas et al. In 2024, they showed that there's a slight improvement with pregnancy rates, clinical pregnancy rates, and live birth with NIPGTA. Although very closely competitive, however, they display that there's a slight improvement with your pregnancy rate and live birth rates with NIPGTA, and the NIPGTA has no impact on the viability of the embryo. So NIPGTA can also be carried out on thawed blastocysts. So the embryo has been frozen, we can thaw the embryo and also still culture over day 5 into day 6, and we can still get this genetic material to be able to get information about the blastocyst. Here at Nordica, we actually are clinically validated to carry out NIPGTA, and we've been authorized to carry out clinical cases. So here we conducted a study on samples collected between 17th of May 2023 to 17th of January 2024. And we carried out this clinical validation where we carried out the conventional PGTA and the non-invasive PGTA. And we had a concordance rate of 87.5. So this authorizes us to also be able to carry out this NIPGTA here at our center in Nordica. Okay, so with NIPGTA, what are the future directions? NIPGTA is here to stay. And so in future, in line with the future directions, we are looking at combining NIPGTA with metabolomics and proteomics of the spent medium to also get more information about the embryos. We're also looking at wider access across Africa, scaling NIPGTA and making it more accessible to patients across Africa as well. AI is actually here to stay, so we will not be completely eliminating the role of AI even with NIPGTA. So we're still looking at prospects of pairing the molecular results from the NIPGTA also with AI morphology for a sharper and more improved embryo selection. The process of NIPGTA also requires multiple washes to ensure that contaminants from maternal or paternal DNA are being eliminated. So hopefully in the future, we're also able to automate this process and make it more seamless. And the regulation and ethical concerns are also valid. However, this is still ongoing, and we're hoping to also make NIPGTA a routine clinical procedure for our patients. In summary, NIPGTA is a major embryo-safe advancement in IVF that enables us to have a true molecular and genetic result of our embryo without the embryo biopsy. No compromise in any way to the embryo, the blastocyst viability, and the pregnancy outcomes are preserved. Here at Nordica, we've been validated to carry out this procedure with an 87.5% concordance recorded from our own research done here. Continuous standardization and validation will secure the place of NIPGTA in routine practice. Together, we can improve IVF outcomes safely, affordably, and ethically. So here are my references for the presentation, and thank you for listening.
Yeah, thank you so much, Toluwanimi. Yeah, and yes, for the beautifully delivered paper on non-invasive PGTAs. Anyway, so I think we'll reserve the questions for when all the presenters are finished, because we're talking about so much technology now. I think that's where Darren comes in. We really need to also look at the ethical considerations in all the genetic testing that we're doing. We're balancing technology with patient care. So I think I'm just going to yield the floor to you, Darren. Please do justice to this. Let's see how we can use our technology with responsibility. Thank you.
Okay, thank you very much. And thank you so much for the invitation. I hope to come and visit your beautiful country one day. And very nice to be giving a webinar that's in the same time zone. I have to say, it's absolutely fantastic. So I'm very, very grateful. Greetings all the way from the UK. So first of all, a few declarations of interest. This is me. Currently where I am is operating as a freelance professor. I do have a 1 day a week at University College London. I was something like 20 years professor of genetics at the University of Kent, and also with a position in Bangkok as well. Various editorial work and some active contracts as well. And if you are interested in genetic consultancy services, that's my website. So please do check it out, griffinlaw.com. Right, so as Lara very beautifully described here, we have about 4 different types of PGT-SR. And I think it's fair to say that 2 of them are very controversial and 2 of them are not. So in the case of PGT-M, as Lara described, there are universal approaches like carrier mapping and haplorhythmasis that are being widely adopted now. When I wrote this slide, there were about 30,000 slides worldwide. There are probably nearly 50,000 now. I'm going to mention 1 or 2 little controversies around it, but by and large, it's an uncontroversial procedure. Compare that, if you will, to PGT-P for polygenic disorders. This is not widely practiced. As far as I'm aware, there's 1 group in the USA doing it. It is in its early stages, and it is very controversial. I'm only mentioning PGT-SR for the means of completeness. I don't think we have many ethical issues surrounding that. One possible one that I might just give the briefest of mention to when I pass it. And then finally, PGT-A for aneuploidy, and formerly known as PGT-S, screening for chromosome abnormalities, preventing genetically affected live births, miscarriages, and IVF failure. And it's fair to say that's still pretty controversial. So let's do the first of those. Now, we heard not so long ago that initially we targeted the disease mutation alone. Then we looked at linked and polymorphic markers, but now we use SNP microarrays, and potentially we might be doing a lot more sequencing in the future. And carrier mapping was an approach that was developed back in 2009, 2010. These were the very first cases. And I'm very proud to say that as a result of just last year, using a single test, one of the things that we've been able to do is diagnose over 1,000 unique disorders. And here you can see just some of the breakdown of those. So cystic fibrosis is still by far the most common. We have Fragile X, sickle cell, and so on. So this demonstrates the wider applicability of carrier mapping, both in its current form, which is with an Illumina SNP chip, but also it can be applied to sequence-based data. So for instance, the PGT complete test. Now, there is no doubt that PGT-M will become more popular as carrier screening increases. And I think it's fair to say that carrier mapping or similar principles like haplorhythmesis will be performed by sequencing, but hopefully by still using haploblock analysis. We've just heard about non-invasive PGT, spent media, blastocentesis, avoiding the need to biopsy. But one of the questions I want to address ethically here is, what about if we just go and sequence the whole genome? Great, surely that's the holy grail of all the things that we want to do. Well, possibly. Because some of the things that we might want to consider is, what about if it finds something that is of unknown or semi-known clinical significance? What if there's just one paper out on this particular mutation in the whole of the in the whole of the literature? What about if it's if it's just one case report, or maybe just one family? And some of the things that we just need to be very, very careful about is, are we challenging the principle that every child has to the right of an open future if we know its genomic code in its entirety right from the outset? The other ethical consideration that we might want to consider is the increased responsibility of the clinician or the genetic counselor towards the welfare of that future child. So although we say, oh, yeah, great, we're going to be able to sequence an embryo using PGTA pretty soon, I think we need to just be a little careful of what we wish for, because this designer babies label is always with us. And one of the advantages of using something like carrier mapping or an adaptation of it is, although it scans the whole genome, it only screens for the disease in question by comparison with affected individuals. So it's just a note of caution there, because when we get onto the realms of PGTP, we get into all sorts of different areas. And I think it's important really just to start off where our definitions are. PGT-M means monogenic. It means literally one gene. So strictly speaking, PGTP is a diagnosis of more than one allele, more than one gene. And if you think about it, in that case, it's already with us. We have digenic syndromes, retinitis pigmentosa, Bardet-Biedl syndrome, certain forms of deafness, Hirschsprung disease, and so on. We have cancer predisposition syndromes, where there is a primary mutation, such as in the APC gene or the BRCA genes, but we might be able to just have a look at a few more genes and say, well, this gives us a better idea of what the penetrance of this primary gene will be. And of course, we have savior siblings. We look at the disease and we also look at the HLA type. So these things are with us already, and we've already taken the first step on the slope of doing PGTP. Thus, for the purposes of this presentation, I'll use the following definition. It's just a personal one that I tend to use. It doesn't necessarily have to be your definition, but if you are looking at detections of variants in 3 or more genes for one single condition, where there's a degree of polygenic risk score information that's being used. What's a polygenic risk score? Essentially, it's a percentage risk that an embryo will develop into a person with a particular polygenic disease based on current evidence. And the way that that evidence has been derived, because many groups have studied large data sets with the disease and performed what we call genome-wide association studies. Now, these are currently being used in adult populations to advise things like lifestyle choices, effects of certain medicines, and so on. But the question that we need to ask ourselves is ethically, should these polygenic risk scores be used on embryos? Here are some of the positives. You're going to rank the embryos anyway, so why not include an element of polygenic risk score at the same time? For variable expressivity and less than 100% penetrance genes, PGTP should not be such a big step. And some people say that, say, for BRCA1 and BRCA2, if we looked at a few more genes, we might get a better idea of the genuine risk of someone developing breast cancer. And we do BRCA1 and BRCA2 for PGT already. Potentially, if you practice it widely, you could reduce the risk of these polygenic multifactorial diseases. If you think about it, monogenic diseases, even in their totality, are about 1 in 50 live births. So that's significant. It's certainly significant if you're a family carrying it, but it's not the big killers like cancer and diabetes and schizophrenia and so on. And they would say, one of the criticisms is that we, different populations, the different ethnicities have different risks and so on. Well, some people would say that that would be already being addressed. A good starting point, some people say, would be diabetes type 1, because actually an increased risk of diabetes type 1 is apparently also associated with a decreased risk of IVF success. So the argument is that PGTP under strict regulation on a case-by-case basis should be no different to innovations that back in the day we found previously found unacceptable. It's Louise Brown's birthday today. Well, at the time in 1978, it was very, very controversial. PGTM was very controversial. It isn't now. And cloning and stem cells and chimera and save your siblings, all of these have been in their time hugely controversial, and now they are widely accepted. On the other hand, you could say, well, this puts us on a slippery slope. I've already said we're already on the first step of that slippery slope. I'll deal with that in the next slide. What about cost? It will only be available for the rich. One of the things that really bugs me as well is that will it encourage complacency? If someone is diagnosed with a low risk for cancer, does that mean then that they are more likely to drink and smoke and have a poor lifestyle because they're safer in the knowledge that they're a low risk group? And hence, do they put themselves in a higher risk group as a result? How do we select the families? Where do we go first time? What about these incidental findings? What about this public backlash? It's inevitable. What about this issue of too much information? And also times change. What is a risk phenotype now may not be in 40 years because of the changing environment. And have we really established risk? Follow-up of cases could take decades. Now, some people said, well, that's a great business model because by that time, all the CEOs of all the businesses that are peddling it will all be long gone and dead. And of course, some of the people will contract the disease even though they were diagnosed as low risk. People don't understand risk, and it's hugely important to make that point. But here's a question. Are any of these reasons why we should not consider it in selected and regulated cases? And this is the ethical dilemma in front of us. Now, I want to go back to this first point, the slippery slope argument. Diallelic syndromes, cancer predisposition, save your siblings already with us. Next step on the slippery slope, monogenic disorders of low penetrance or variable expressivity, like our BRCA1s and BRCA2s. I've mentioned those already. Then maybe selected high-risk families for specific polygenic diseases. We talked about diabetes, for instance, or maybe as a tool to prioritize the transfer of our remaining euploid embryos. All of these, I would argue, are things that we might want to consider. However, that is an awful lot of work. So why do we need to go further down the slippery slope just yet? Screening of the polygenic diseases is most concerned because that's the next stage. Or the next stage, more widespread routine use, whole sequencing with as much information about disease risk. Or right at the very bottom of a slippery slope here, routine screening for multiple traits, including our cosmetic ones, are real old designer babies here. So my argument is that we've got a lot of work in these 2, 3, and 4 here. So why not concentrate all the brain trust and all the effort and really just say, look, we're just not going to go here for 5, 6, and 7 for a while. Because the considerations that we need to give ourselves is how pointless or worthwhile is it? When we've eliminated the aneuploids, the grossly abnormal embryos, how many are left to look at polygenic risk score? And really, how much will each embryo be different from the next? And Tony Gordon and I wrote this paper called Mad Pursuit, and it's a Francis Crick quote, and it makes the point is that what mad pursuit will we be in a situation where we're agonizing over a PRS of 55 versus 50%, when we really should be focusing our attentions on how likely is the embryo to implant and lead to a live birth at all? Now, I mentioned that I'm not going to talk about PGTSR. I think the only ethical consideration that sometimes comes up is do we screen for carrier individuals like their parents or just affected ones? I'll leave that one open because I want to focus really on PGTA. Aneuploidy, the A in PGTA, extra or missing chromosomes, causes all of these disorders. So ethically, PGTA should be a no-brainer. Just select the euploid ones. But it's not. A lot of people argue against it. Who is it for? Well, there's your referral categories here. And there are a number of issues. We have issues like, can biopsy damage the embryo? Well, not if you're using non-invasive. And some people will say, well, if you do it right, you don't damage it. But I am going to deal with some of these issues here. Stubborn people, including biostatisticians, how we did interpret the data, and then my favorite topic of mosaicism at the end. So can biopsy damage the embryo? Yes, it can, but it shouldn't if you do it right, is the short answer there. Let's get on to our stubborn people now. A book to read before you die, ladies and gentlemen. This is Win Bigly by Scott Adams. Scott Adams recently died, sadly, but he was the author of the Dilbert cartoons in newspapers in the US. And he really talks about, the whole book is about how Donald Trump managed to win his first election, and how he managed to persuade people in the absence of facts, essentially. And the reason is this, that by and large, he can really, really tap into a trait or a set of traits that we all have. And as scientists, the sorts of things that we should fight against, the sort of thing that the scientific method and our scientific training fights against. And the first is cognitive dissonance. So dissonance just means, oh, I can't be bothered with that sort of thing. But cognitive dissonance is you do the, oh, I can't be bothered with that thing, but you're still nonetheless aware of the risk. You're cognitive of it. So a lot of people do this with smoking, for instance. Oh, well, you know, I had an Uncle Norman who smoked 40 cigarettes a day, and he was all right, so I'm probably going to be okay, regardless of the rest of the evidence. I sometimes do that when I drink too much, I have to say. So I've got that a little bit as well. The second one is confirmation bias. And again, this is one of the things that's becoming more and more difficult to be able to try and get away from. Because confirmation bias is just picking the bits of evidence that we want to believe in order to bolster our own points of view. And there's a lot more evidence out there. Now, the point I'm going to make here is on both sides of the PGTA argument, there's increasing evidence that when we see a paper either for or against PGTA, that tends to polarize opinion rather than find consensus. And sadly, too many people have made a career out of being very pro or very against. So it's a little bit of a tongue-in-cheek exercise. Myself and Sally Sheldon, who was the professor of law at University of Kent when I was there, did a little sort of slightly humorous approach, and in which we invented two characters. Now, the only thing that's true about these two characters is their names, and they really are called Jacob and Giuseppe. Everything else I'm going to tell you about is complete fabrication, but it makes this point. So in this imaginary world, Jacob is against PGTA. His mantra is evidence-based medicine, and his modus operandi is more complex analyses need to be done before any new treatment is put into the clinic, and the result is that very few do. And his worst fault is confirmation bias. He's selective about the evidence that supports his own point of view. Similarly, Giuseppe, in this imaginary world, says, I will always do what I think is best by my patients. He's motivated by keeping his IVF clinic open, by the business, keeping his staff in gainful employment. And of course, his worst fault is he's selective about the evidence that supports his point of view, confirmation bias. Now, you'd sort of hope that when in a normal free-thinking society, we would have a range of views. Of course we would, with Jacob and Giuseppe at the extremes, but with the majority somewhere in the middle. And when we saw a degree of empirical evidence against PGTA, we might move a little bit more towards Jacob. And if we saw empirical evidence supporting PGTA, we might move a little bit more towards Giuseppe. Unfortunately, what tends to happen, this is not an empirical study that I've done, it's just what I observe, is that when you see evidence about PGTA, be it positive or negative, as I said, it tends to polarize opinion, and more people just dig in their heels, unfortunately. So let's just turn our attention then into how we interpret the data, because what I want to turn your attention to are randomized controlled trials. This is the most recent one, possibly one of the most famous, and in a very, very long nutshell, it says that PGTA does not work. If you look really not that closely into this paper, one of the things you find are the following. The patients are of good prognosis. They're on average 25 years old. This study only measures cumulative live birth rate. And sadly, there are too many of these studies around. And this is just an example of more stubborn people. PGTA was never there to improve cumulative live birth rate. If you think about it, it can't, in theory, because you have a pool of embryos and eventually you're going to pick the good one. PGTA just gets you there quicker and it avoids miscarriage. So a study that I'd just like to point your attention to, this is one that myself and Santiago Monet put together relatively recently. And it was a very, very simple study indeed. And all we did was we looked at the SART database and we asked the following. What is the live birth rate per embryo transfer? And 4 things you get from this graph. First of all, the with PGT is higher than the without. Okay, so it's showing an effect. The second thing is the brown line without PGTA is very steep, so it shows an effect of maternal age far more than does the with PGT. Now, the one with PGTA does still have a maternal effect, but it's much less pronounced. The third thing is even at the younger age groups, we see a difference. And then the final thing is that without PGTA, the live birth rate per embryo transfer is the same as if you're the age of 42 with PGTA. So in a nutshell, is PGTA effective in improving live birth rate? For cumulative live birth rate, no. For live birth rate per treatment cycle, maybe. And for live birth rate per embryo transfer, definitely yes. And these are the things that we need to be very, very careful that when someone starts jumping into an ethical argument, they will jump into the bit of evidence that they want to see in order to bolster what they think is an ethical argument. But the important thing about ethical arguments is you need to start with the facts in the first place. It also reduces the chances of pregnancy loss. Now, I think it's got to the stage now where almost everyone agrees that this is a real effect. And here you see it again. So it's almost like the reciprocal of the graph that I've just shown you. The brown bar going up and steeply, the blue bar staying relatively level associated with maternal age, and then the under 35s with and the over 40, sorry, the under 35s without and the over 42s with are about the same. And hot off the press, preterm birth. We see a difference there as well. So I would say that if you're in a position where you have to deal with patients, and you have a young, good prognosis patients who are unfazed by repeated attempts of PGTA, then it's not for them. Because PGTA is and always has been designed for its referral categories: advanced maternal age, recurrent pregnancy loss, recurrent implantation failure, male factor, infertility. All patients ethically needs to know the facts to empower them to make their own decisions. But importantly, it was never been there to design, never been there and designed to improve cumulative pregnancy rate. Now, we did hear from Lara recently, and we've also published a paper on this as well, that it's now being used to look at the non-2PN embryos and the poor quality embryos. So maybe that's creating a new pool of embryos that might, sometime in the future, improve cumulative live birth rate. And then we change the argument, but not until then. Because what happens if we transfer an embryo that is diagnosed as euploid? The short answer is the chances of a biochemical pregnancy are twice as much with a euploid versus an aneuploid embryo, 3 times as much to get a clinical pregnancy, and importantly, the live birth rate when you transfer an embryo that is completely aneuploid, so 5 out of your 5 cells in your biopsy are abnormal, your live birth rate is 0. So, with a euploid embryo, 65% chance of live birth or an 8% chance of miscarriage or 9% of the established pregnancies. With an aneuploid embryo, let's be generous and say 1%. It's probably nearer 0, but a 3-fold increase in the chance of miscarriage, or indeed 100% of the established pregnancies. So the dilemma. So in the next graphic, I'm going to show you euploid embryos in green and aneuploid embryos in red. The dilemma of a prospective PGTA patient ethically is this: if, say, for the sake of demonstration, they have 5 embryos, if they're 25 years old, then maybe 1 of them is aneuploid and 4 of them are euploid. But that's going to go down. But with PGTA, they are empowered with this knowledge. Without PGTA, every embryo is gray. We don't know. And some people want to know that. And that's the crux of the ethical argument, if you like. And you might say, well, hang on a minute. Didn't the UK regulator, the HFEA, the Human Fertilization Embryo Authority, give PGTA a red light? And in fact, yes, it did. It gave it 2. It had this traffic light system for IVF add-ons. But here's the thing. In point of fact, when they had their traffic light system, they didn't award any green lights, and they didn't have a provision for green lights to be awarded. 1 or 2 of the committee members were coming out with phrases like, "There's no evidence that will convince me that PGTA is effective." They didn't acknowledge that graph that I showed you just a moment ago, which is from a non-selection trial, saying that there is no evidence that is effective, which is what they did, is factually incorrect. What they should have said was no evidence for randomized controlled trials that it improves cumulative live birth rate in all IVF cycles. And that was their threshold criteria. Now, importantly, as I mentioned, an approach designed to select the best embryos will only ever get a red light. Now, of course, and they also license its use. So on another part of their website, this is a licensed and usable procedure. So all these things I argued with them was not a good look. And they very graciously invited me. They listened to me. When I spoke to them, they removed at least 1 of the red lights, and now their guidance is very different. So it looks a little bit like traffic lights now, but it is somewhat different. And they were wonderfully gracious. So one of the things I would say to you that everyone at least agrees now that improves miscarriage rates. So my last few slides now are just on my favorite topic of mosaicism. And one of the things I'd say to you is just take a little bit of a chill pill here and think about it. Your cell, your blastocyst, has about 200, your embryo, sorry, your blastocyst has about 200 cells in it. You're taking 5 cells out of there. Okay. If all your 5 cells are chromosomally the same, You don't have a Scooby-Doo. You don't have a clue about whether the embryo is mosaic or not. Your diagnosis is of your biopsy, not of your embryo. And your biopsy result can only ever, at best, be a reasonable proxy for the mosaicism status of your embryo. But we've modeled this, and what we think is it's still good for ranking embryos nonetheless. So it is important. Now, I'm going to say this loud and proud. I've been doing this for an awful lot of years, over 35. Most of the evidence from most of the studies, and I think every human embryo is mosaic. Okay. All the cells could be abnormal, but they're not uniformly normal. They could be chaotic. They could be, a million different abnormalities in your embryo. But I challenge you to take apart an embryo, a blastocyst, cell by cell, and not find one that is chromosomally different to the rest. Now, it could be that the ones that we're worried about are euploid, aneuploid mosaics, where some are normal and some are abnormal. And one of the things that I would caution you is don't just imagine that's the only form of mosaicism. Mosaic means that not all the cells are uniformly the same. Now, we do know that we see more abnormalities at day 3 than day 5. But one of the things that's hugely important is the human embryo is karyotypically diverse and it's fluid and it's dynamic. So we learn in our textbooks that a sperm with 23 chromosomes meets an egg with 23 chromosomes, and they fuse to form a zygote with 46 chromosomes. And it's 46 chromosomes thereafter, unless there's a mistake, unless there's an error. I would say to you, think the other way around. Think backwards, because in point of fact, every embryo has got at least one abnormal cell in it. Which gets us back to our patients then. What do our patients want to know? And does PGTA help? Will I get pregnant eventually? Well, that's our live birth rate, either per treatment cycle or our cumulative live birth rate. So that's the subject of some debate. But will this transfer lead to live birth rate, to a live birth? That's our live birth rate per embryo transfer. Yes, it will. Will I suffer pregnancy loss? That's our miscarriage rate. Does PGTA help? Yes, it does. How soon will I get pregnant? That's your time to first pregnancy. Does PGTA help? Yes, it does. Cost is going to vary on the clinic, on the package that it offers. But if you think that roughly a PGTA is, depending on the clinic, depending on its business model, is roughly the price of an embryo transfer, then you can see roughly where the balance occurs. If you have more transfers than you do PGTAs, then you're in negative balance. And then, of course, we need to consider that these are not figures on graphs. These are individual patients, each with their own specific journey. And how satisfied they are is very much dependent on their experience and their personal circumstances. So, for instance, if they only had one embryo, now, doing PGTA is not going to improve their success rate, but empowering them to know whether that embryo is aneuploid or euploid may just be very, very good for them and their experience. And do please just bear that in mind. And so this is a paper that we submitted, myself and 2 genetic counselors recently, about a decision tree. I think all patients should be made aware of PGTA. Okay. Once they're made aware of all the risks and benefits, and they prefer, no, I'm going to transfer all my embryos if necessary, then PGTA is not for them. What are the things they want to consider? Live birth rate, pregnancy per treatment cycle, pregnancy per embryo transfer, miscarriage, and so on. They might be of a lower egg age. So what I mean by that is they themselves might have a low maternal age, or they might be donors. Reduced risk of aneuploidy. Well, they then need to do a risk-benefit analysis. Similarly, they might be over the age of 35. But in each of those cases, they may or may not choose to do PGTA. And ethically, this is the thing that we always need to consider. It comes down to each time this is someone on their own reproductive journey. And when we start showing graphs and statistics and so on, we need not to forget that. So here I'm going to conclude now with a few. I gave this talk in Athens recently, so hence a slightly Greek theme. Myths and truths. First myth is PGTM requires development of one test at a time. Now it doesn't. We've got universal PGTM around there. PGTP is all about cosmetic tests. Well, no, there are genuinely ethically sound areas of PGTP that might be worth considering if we regulate it properly. Embryo biopsy damages the embryo. It can, but it shouldn't if you do it right. PGTP cannot improve cumulative live birth rate. Well, no, at the moment it doesn't, and I've said it doesn't. But recent evidence, if we look at the non-2PNs and the poor quality embryos, suggests that it might do in the future. A biopsy result is an accurate representation of the mosaicism state of the embryo. No, it's a reasonable proxy. It's a good way to rank your embryos, but it's not necessarily chapter and verse. A human embryo is usually chromosomally uniform. No, it's not. Probably all embryos, even the aneuploid ones, are mosaic. Okay, you can't transfer mosaic embryos. You can. If you have a mitotic error, and it shows you have some normal, some abnormal cells, the evidence suggests that you can transfer embryos. And with that, I'm just going to recommend my book and my website. So I will end there. Thank you very much.
Yeah, thank you so much, Darren, as usual. Yeah, thought-provoking. I think we just go to the questions now so that we can take all the questions. I think there are a few of them. Maybe I'll start with Laura, and I think you mentioned something about the workflow where you have time-lapse and PGTA together. Actually, it's one of the questions that somebody also asked. I mean, how the synergy between these two, can you just describe the synergy between these two?
Yeah, absolutely. Let's start in a sort of, I would say, maybe chronological order, starting from fertilization, for example. We put together all these evidence regarding and time lapse, and obviously one of the important benefit of time lapse is to have a greater pool of embryo documented as 2PN, because the observation is very precise. You can go back and classify more accurately your embryos. And this is the same for a typical fertilization, so 1PN, 2.1, or 3PN embryos. And the combination with precise pronuclear observation and PGT with ploidy analysis can really improve not only the pool of embryos, but also the number of embryos that are euploid and diploid, and it can be used and saved and rescued. And we have been able actually to prove these better pronuclear observation. You can even go back to the time lapse and confirm in some way the results that you have with PGTA. So, for example, you have a triploid embryo, you can go back and see if it is a real non-extrusion of the second polar body, for example. So, sort of synergism in confirmation of the results connected with the pronuclear status. This is the first, I would say, step. Then if we talk about cleavage stage, we know that morphokinetic can predict the blastulation. This is important not only from the IVF lab perspective for workload planning, but also to have in mind the number of blastocysts that your patient can potentially have. And I see this as a synergy also with PGT, because it's particularly relevant for those cases where the number of embryos available is reduced, for example, for a monogenic condition, for a translocation. So to be able to counsel your patient and setting their expectation, having both time lapse and PGT can make the difference, because you can again better counsel your patient and say probably you will need an additional cycle, because your probability of having a euploid and normal embryo is reduced. So this is another, for me, clear point of synergies. And the most important is at the blastocyst stage. So when we talk about biopsy and also just simply talk about the quality and quantity of cells that we pick up with the biopsy at the blastocyst stage that we have then to send to the genetic lab. Obviously, time lapse allows you to visualize the embryo, to have good quality embryo with clear ICM, with clear trophectoderm. So we are really taking those good quality embryos, which is mandatory then to have a genetic result. So if you want to have an informative results, you need to provide a biopsy that is of enough good quality and quantity. So, and also underpinning principle of getting good quality biopsy is, for example, undisturbed culture provided by time-lapse. So I see these synergies at different levels. These are just, I would say, 2-3 examples, but we can have even more.
Can I just ask a more difficult question and ask you if I wanted one of these two methods in the laboratory, which one will you advise me to get? Is it PGTA or is it time-lapse?
I mean, is a good question. I think that independently they have both a lot of value because, I mean, time lapse for all the reasons that I mentioned, but even independently as a tool to improve the culture system, the way of observing the PNs. So I would not put them necessarily in competition. I see them both as in an integrated system. There is no need to say time-lapse and morphokinetic algorithm are better than PGTA in giving, euploidy and aneuploidy. So as a combination of the two tools. So ideally, if you can have both, I would say even better.
Okay.
I think there's no reason not to have time lapse for all of your embryos in some ways, because you have undisturbed culture. You have one of the things with the patient journey is they get to see their own embryos, which is lovely. And as a means of diagnosing chromosome disorders, then of course PGTA is better, but it is only for your referral categories, whereas the overall experience for a time-lapse system has a number of added advantages as well. I think that's a thing to bear in mind.
Wonderful. Yeah, I listen to every one of you. One topic that is not coming up is autism. And each time we talk about genetics, parents want to hear about autism.
Really?
Is there anything about autism? Where are we on autism?
Well, I'm blessed with autism, so I'm not a big fan of screening for it. I wouldn't exist.
So you'll have been screened.
I've been tested. I don't know whether there's a genetic test for it, but yeah. And I think you'd wipe out 50% of science departments, 90% of computer science departments, and I know that the autistic community doesn't like, you described as being on a spectrum, but you don't like to be described as how far you are on that spectrum anymore. They don't like to put a number on it. So I think you go down a line that is, ethically very fraught in some ways.
Okay.
Yeah, but Darren, did you read those evidence that advanced paternal age is increasing the probability of having autism in the offspring? So I've been requested more than one time if it is appropriate also to talk about this, because we are always focused on the mother, but also the father is important and is contributing in these kind of things with de novo mutations and again increasing paternal age. So yeah, just as a consideration, because we typically target just
segmental aneuploidy. So there you go.
Yeah, exactly.
Yes, there are a lot of questions about non-invasive PGTA, so I'm going to come to them last. So, ethical consideration, is it right for the patient? Is it the right of the patient to know about the disease risk and his decision to seek management or watch and see? I seriously, I think I don't really understand the question, so I don't know whether you guys will understand the question, but I guess that's for Darren anyway, but I don't understand this.
Is it the right of the patient to know?
Yes.
I mean, if they've gone into a clinic for the test already,
Mm,
then yes, of course, I would say. Um, I mean, the thing that worries me is what if you find something else?
mm,
And you know how, and particularly if you are you know if you're unsure what it means. So certainly, for instance, when we do, in many ways, this is a little bit with us because when you do a test for a monogenic disorder, sometimes you will do a PGTA at the same time. And the PGTA is getting so sensitive now, you're spotting microdeletions, and we don't necessarily know the clinical significance of every microdeletion that we've ever seen. So I still think it's worth telling the patient, but you know that there's a real skill in genetic counseling in how much you tell them and at what time.
mm,
And you know it isn't an easy solution, I would say.
mm. Thank you. Yeah. Now, Laura, let me come back to you on training in genetic counseling. Is there anywhere now that we could, especially maybe let's take Europe together, and of course, I still take UK as part of Europe. Is there any place where you could have training for genetic counseling?
Yes, there are many. If you look at the website of the European Society of Human Genetics, they have the Board of Genetic Counselor, and they are a list of universities which are sort of linked to the European Board and which are accredited and licensed to train genetic counselor. We have also to mention that, I mean, I'm working with many countries and the regulation is different in different countries. I work with Middle East, for example, and every country has its own license and own way of training in terms of genetic counseling. But at the European level, if you go into the website, there is specifically a list of each university with the programs that they have and that they offer for genetic counseling. Then you have to understand at the local level if this is recognized or not as I would say as a position. In Italy, no. For example, so genetic counseling must be performed by medical doctors or other type of roles, but with a specific school. So again, at the European level, there are many opportunities, but then check with local regulation, at the local level, what is possible and what is not possible to do. This is my suggestion.
Okay, thank you. Okay, one more question before I go to the NIPT because there are quite a number of this. Does IVF rule out chances of genetic disease like leukemia, sickle cell, or any of such? Is it possible to rate it? So actually, I think it's talking more of PGT-M. Can we screen for, and probably PGT-P, can we screen for diseases like, of course, I think the answer should be yes. Sickle cell, we already, we're doing that.
Sickle cell is routine, yeah.
Yeah.
Certain form of leukemias, inherited leukemias will be testable. But most leukemia is an acquired abnormality, so yeah, you're not really able to.
Okay. Yeah. Thank you. Yes. So, what is non-invasive PGTA possible in a co-culture mechanism? You are the embryologist, so.
Okay. Well, yes, up until day 4, but after day 4, the culture would have to be individualized because the genetic material usually would start to be expressed in the spent culture media from day 4. So co-culture can happen until day 4, but after day 4, individualized culture.
Okay. There's still one more question for Darren, but let me try to see some of this. Do you think NIPGT has the potential to replace invasive embryo biopsy completely? Or will it continue to remain complementary to the conventional?
Conventional. Well, NIPGT can be used in routine clinical practice, but in terms of replacing the conventional PGTA, I think the indication should be taken into consideration. So the indication for doing the PGTA would determine that.
Okay, thank you. Thank you for the insightful presentation. Since non-invasive PGTA using spent culture media has already been implemented at Nordica, could you share the minimum volume of spent media required for reliable analysis? Whether the technique can be performed using sequential culture media as effectively as single-step media, and based on your experience, whether you believe it is ready for routine clinical practice or still better used alongside conventional trophectoderm biopsy. There are many questions in one.
Exactly. All right. So for the volume of the media, usually the embryos are being cultured in between 10 to 20 microliters of the media. And so usually that's about what we would need for the analysis. In terms of single step and sequential, I think single step, particularly if utilizing the time lapse, but I think you could also use sequential media as well.
Okay.
So sequential because from day 4, you're also going to be changing the culture of the embryos. So I think either works.
Okay.
And then the final part of the question.
Yes.
Please can you think that again, sir?
Yeah, whether you believe it is ready for routine clinical practice. Is it ready for routine clinical practice?
Well, so far so good. Yes, we can, but studies are still ongoing and improvements are still on the way. But I think it can be utilized for routine clinical practice, but if there's a clear-cut indication to do that.
Okay. All right. So the indication must be there. Okay, fantastic. Now, this is probably for Darren because it's an ethical question. How do you handle the dilemma of advanced maternal age? A woman not willing to accept donor eggs, what options are open to such a person?
If she doesn't want donor eggs, then yeah, I mean it's advising about PGTA is pretty much the only option at the moment. But also a realistic a realistic counseling approach. Her ovarian reserve may also be low. She may only have 1 or 2 embryos in the first place. How many times does she want to go through stimulation cycles of producing 1 or 2 eggs, and in the hope that you eventually get a euploid one? So a degree of reality in that. Looking forward, I know that there are companies that are trying to develop means of improving egg aneuploidy. There's a company called Euploid, which are doing that. So we're all looking forward with bated breath about how successful they might be sometime in the future. But it's a long way away, I would say.
Yeah, there's something coming up, I think, especially, I mean, in the literature, but more common in probably in Nigeria and other developing countries, this PRP thing. And a lot of people you see 48-year-olds saying they want to do PRP to improve eggs, the quality of their eggs. What's your take on this?
I don't know enough about it. I mean, what is platelet-rich plasma, isn't it?
Plasma. Yeah, that's right.
And I don't know. I wouldn't say I've seen a huge amount of evidence to say that it improves egg aneuploidy. I know that in some, I'm just looking at a website from Johns Hopkins University, for instance, wound healing and trauma and joint injury, all those sorts of areas. Reproductive medicine, I don't know yet, but I don't know. Who knows in the future?
Yeah. Laura, do you have any take on this as well?
No, I mean, it's difficult to have a treatment that is able to cure aneuploidy or to remove the burden of aneuploidies in female age, unless it's something that is going to act at a molecular level. So you have to really act on the DNA, I guess, to remove these, I mean, affected chromosomes. Otherwise, I mean, potentially the quality, but talking about aneuploidies, I mean, I'm not getting the mechanism for which they can correct an aneuploidy, honestly.
Well, Tolu Anime, do you want to have a go at this? Any opinion?
No.
Yeah. I think we just need to be able to give correct counseling to the patients and accurate counseling, give them enough information. I think so many people are trying to give this hope, where sometimes it is not really, there's nothing to back it up. So, but I'm sure some clinicians are listening. And then, because we are getting a lot of requests now, even 50-year-olds are asking for PRP. Anyway, and I guess some people are giving it to them. Anyway, I think it's been wonderful having you guys around. As usual, it's been a wonderful conversation. So many of our people have given very positive reviews about the lectures and everything, and it's always good to have these conversations. Thank you so very much, Laura, for making time to be here today. Tolu Anime, it's been wonderful having your lecture. And Darren, always a pleasure to hear you. Thank you so very much for...
It's been a pleasure as well, yeah.
Yeah, and about coming to Nigeria, we'll talk about it. I think one of these days we should have you around.
Okay.
Thank you so very much, and have a lovely evening.
And you got to invite Laura as well, you know?
Oh yeah. Laura, she's in Italy. That's one day we're going to talk about it. Yeah, thank you so very much. And
Thank you.
See you guys.
Excellent. Cheers, guys. Take care.
Thank you. And all of you for participating.
Bye.
We're so grateful to you guys. Thanks.
Yeah, bye.
Bye. Yeah.